Good morning, friends out in YouTube land. Stop. So, we're in our last week here, and what we're going to do is begin placing cells into a bigger context this week. Putting cells together, making epithelial sheets, and additionally, placing cells into the extracellular matrix, the surface that they're laying on top of. And this then begins to tie into some of the mechanisms of cancer, that we have actually been covering a lot of things in terms of basic molecular genetics and particularly cell signaling. But now, the way cells can behave to form a tumor, you have to have an understanding of how cells are interacting with each other and how they're interacting with the environment around them. So it's a really interesting topic, and it is our last thing to be hit, and it just sort of opens up more interesting ideas and concepts, but I think that's what we are always doing. So we begin thinking about the extracellular matrix. We've already kind of got the idea that a cell is not just a water balloon filled with jello, or a jello-filled balloon, but we have a lot of structure inside the cell. Additionally, there are connections between cells and connections between the cells and material that they have secreted or other cells have secreted that they can sit upon or actually have on top of them. And we're going to start, actually, in rats. I should grab more markers. Oh, well, we'll see how we go with this. Actually, a little bit of discussion of the plant cell wall, in part because plants get shortchanged around here. We don't do enough plant biochemistry. And additionally, it actually provides an interesting comparison when we look at plant cell wall and extracellular matrix versus animal cell, not necessarily wall, but the equivalent, the extracellular matrix. So plant cells are surrounded by the cellulose, primarily, cell wall. And this gives plants some interesting opportunities in that they can actually have osmotic pressure applied to them. That is, they can have a lot more salts inside the cell than the surrounding water around their roots. They take water up. And if you were to do that to the same extent that plants do, we would end up actually causing our cells to burst. But with plants, because they're embedded in this cardboard box, literally, they actually pressurize. And so we now have a balloon that is, in effect, in a shoebox. And that allows for the plant to pressurize against the cell wall and actually apply pressure, which is why little droopy plants that if you don't water them, they flop over, and then when you water them, whoop, they stand up all happy. And I've given up my mission to try and alter the thumbnail that introduced the video, and now I accept that all thumbnails are me doing this. So go ahead, do it now. All right, so the plant cell wall is a really tough box, made of cellulose primarily. And as we're going to start seeing when we compare with animals, plants are always big on carbohydrate. They have lots of carbohydrate because they're taking carbon dioxide from the atmosphere, fixing it into glucose. And so carbohydrate is pretty cheap. What they don't have is tons of protein, and actually it comes down to tons of nitrogen, allowing them to have an unlimited supply of protein, or at least protein becomes very limiting, which is why if you want your plants to grow nice and happy, you give them a big dose of nitrogen, and whoosh, off they go. So plants don't depend on nitrogen and proteins. They depend on being able to do photosynthesis. That's how they make the cell wall. And so the cell wall, probably not as a surprise to anybody, but the cell wall is made up of fibers of the glucose polymer cellulose. There are very long cellulose fibers And then there is another material very similar called hemicellulose, which is a little bit shorter fibers that get cross-linked in. Additionally, you end up with these bars of cellulose, essentially. And in order to fill in the bars, plants produce yet another carbohydrate known as pectin to kind of fill in the gaps between the cell wall. Pectin, just for those of you that are complete nerds, is a not a glucose polymer, but it is polygalacturonic acid. And if you really want to know the structure, look it up. It is a sugar polymer, it's just not glucose. Polygalacturonic acid is a little bit more soluble than cellulose. And so you know pectin by taking an orange and yanking it open, and all that white material, that's all pectin. So you also know part of what it's doing in that you can take pectin, heat it up, get it to solubilize, so you got to get it near boiling, and then allow it to cool, and bada bing, once again, our old friend of the intramolecular hydrogen bonding forms, and we form a gel. This is how you make jams and jellies. And so if you're using a fruit, a material that's got a lot of pectin in it, like orange peels or various kinds of pectin-y rich materials, you boil it up, add a lot of sugar, which also helps to cross-link, allow it to cool, and you've made jelly. Now, if you're using something like a juice, grape juice, apple juice, you have to add pectin because there's not enough pectin in there for it to make it actually gel. So the plant cell is secreting cellulose, essentially like a little toothpaste tubes coming out and laying down these rows of cellulose, and they lay it down in a very specific way depending on the cell type. And you can get some very interesting shapes in that you can have cellulose fibers that are forming a circle. You have cellulose fibers that are forming bands or lanes and lines. And what this can do is as the cell pressurizes, as osmotic pressure changes, the cell will push up against the cell wall, and depending on how the cellulose is laid down, it may actually change shape. And so there is a specific cell organization known as the stomata on the surface of a leaf. And if maybe in first biology class or high school, you've done leaf peels where you, under a microscope, you can see these stomata. And they look like a set of sort of kidney beans lined up to each other. It's not a great drawing, but close enough. And they've got cellulose wrappings such that when they increase their osmotic pressure, the stomata will actually, the cells surrounding it, will bend to sort of form this kind of structure. And you can actually demonstrate this with literally with a balloon. If you tape it up right, you can end up with a cell that will form essentially a C or a comma on either side, and you have a hole. And this is where you now have gas exchange. And when the plants are photosynthesizing, the stomata open up to allow carbon dioxide in. When the plants aren't photosynthesizing, they can close back up. And it happens strictly because of osmotic pressure. Now, the next question is, how does that osmotic pressure actually happen? Very often with things like the equivalent of a sodium-potassium ATPase, in that you can begin pumping salts ions against their gradient into a cell, and then when the salt comes in, the water follows, pressurizing the cell to another level, and then if you need to close them back up, you begin pumping or allowing to diffuse out the material. Another thing that cellulose is actually good at is that you can cause the cellulose fibers to actually slide along each other by changing the pH. If you dump some hydrogen ions out into the cell wall, all of a sudden they start to slide as the hydrogen ions are affecting the overall hydrogen bonding going on, and you can make the plant actually start to have a cell wall that is no longer stuck and rigid, but now that is slightly flexible. And so if you've ever observed a plant, like a sunflower, that is actually tracking along with the sun, or actually Charles Darwin's son did this, S-O-N, and you can take a plant, expose it to light like a little seedling, and they'll bend towards the light. And he took a little cube of agar on top of the plant on one side of this little seedling and exposed it to light, took that agar tip up, placed it on another cut little seedling, and just the act of putting that on there caused it to bend in the direction of the agar cube. And what he was doing is seeing that there was specific plant biochemicals that were being secreted up into the cube on the side that's turning, that's causing cell wall loosening. And so we actually know a whole lot about how plant cells can track the sun, because they are able to actually recognize where sun is, secrete either different plant hormones or increase hydrogen ions, and cause the cell wall to begin sliding. The turgor pressure begins to push things, and so by balancing these different kinds of issues of pumping salts back and forth and changing the sliding ability of the cell wall, we have plants that can move around and track the sun. It's actually a really cool thing, phenomenon to watch. So that's plant cell walls. And let's see. So there's pectin, which is sort of the filler. Another filler that perhaps you've heard of is a molecule, another carbohydrate known as lignin. Lignin is very, very insoluble. It's like cardboard has got a whole bunch of lignin in it. It's one of the things that when you start dealing with plants, the tougher the plant is, the more lignin it probably has. And like woody species end up having a whole lot of lignin. One of the games in the biofuel biz has been to develop something known as cellulosic ethanol, which is actually a really cool trick. It doesn't work real well, at least I'm saying that on YouTube. I believe it doesn't work real well, in that the idea is when you're making ethanol, the way we typically make ethanol is you take sugar or a sugary juice of some kind. Starch will work, but sugar is better, like cane sugar, and you allow it to ferment. So you add yeast, and you let it burble in the absence of oxygen, and you end up generating a fair amount of ethanol. Ethanol is a toxic byproduct, unlike water and water and carbon dioxide, which is what the yeast would do if you added a whole bunch of oxygen. They'll generate ethanol, and they'll go up to about, depending on the yeast strain, maybe 10% ethanol, maybe if you really push it, 15, 18% would be like way, way high. In order to make ethanol for burning in a car, you have to distill it, and that takes a fair amount of energy. But one of the ideas has been, what if you could take something like switchgrass, just straw, and use that? It doesn't have a whole lot of free glucose. It's got a ton of cellulose. Is there a way you could take the cellulose, break it down into free glucose, and essentially it's the enzyme cellulase, and you then take cell walls, turn that into fermentable sugars, and wow, you'd really be off to the races, because you could take vast swaths of just prairie grass and turn that into ethanol. And it's a brilliant idea. It hasn't worked very well. Not surprisingly, plant cell walls are pretty darn tough. Wood is a pretty serious material to break apart. The extra amount of lignin makes it difficult. If you can, through genetic modification, make a plant cell wall that's a little bit easier to digest, it also ends up being a lot easier to flop over, and plants don't grow very well. So it's a really cool idea, but it hasn't quite come about. That said, just to make a little shout out to fungi, fungi do not have cellulose cell walls. They have cell walls, but they're made up of chitin, which is another carbohydrate polymer, the same thing that's found in shrimp cells, or shrimp shells, and in insect cuticle, chitin. And fungi, they do secrete cellulase, the enzyme that breaks down cellulose, and they are responsible for breaking apart and returning the carbon in that cell wall back into a fermentable or energizable sugar in the forest. And so if it weren't for all our fungi friends, forests would be stacked to the ceiling, whatever, with logs. But logs slowly begin to break down because of the act of various fungi. It's slow going, but it definitely does work. All right, so that was plant cell walls, and I highly suggest, if you're interested, a class in plant physiology. Plant biochemistry is awfully cool, but be that as it may. We're now going to turn our attention to animal cells, which we've been focusing on a good bit more, and looking at the extracellular matrix in an animal cell. So, when we look at tissues, and not so much maybe a muscle tissue, but if you were to take and do a fine dissection of some dude's colon, and you got to have a microscope. So this is something that it comes about, in the late 1700s. This ability to really begin to do tissue histology. We see a really important kind of organization with the animal cells like this, in which we'll have, say, like a gut epithelium. We got the microvilli on top of the gut cell, and there's another gut cell with its microvilli. Remember, there's actin sticking out here and tubulin sticking out here, causing these cytoplasmic projections. And these cells are then laid on top of material that's got a lot of kind of stripy looking pattern to it, a whole bunch of material that it's setting on. I mean, in fact, you can probably observe this yourself if you ever start cutting on a piece of meat. There is membrane. Like if you are fond of ribs, I can't say that I am, but I do know that when you get your ribs, they will often have a layer of this fairly tough, almost like a plastic, that you have to take a knife and get going and then peel it up. I was actually skinning a little bit of fish last night, thinking about exactly this thing. This is what you're doing, is you're peeling away this layer. And so, in this specific case, the layer of material that the cells are sitting on is referred to by a couple of different names. It is known as the basal lamina. The basal lamina is a term a fairly old-fashioned term. Another term for this is the basement membrane, for obvious reasons. Membrane. And the general collective term for this material is, of course, what we call the old ECM, or extracellular matrix. And we've got a basal lamina that is pretty obvious to see, but cells also, depending on the cell type, they'll have material that they'll secrete up here, have sort of an apical matrix or an apical lamina rather than just a basement and a basal stuff as well. So material is being secreted out of the cell. And just to sort of throw back and connect, how do proteins get secreted out of the cell? Via the Golgi. Remember, we've got the signal sequence that is targeting a ribosome, an RNA, into the ER. The protein is being co-translationally translocated into the ER. It then goes into the Golgi. And if there is a transmembrane sequence, like for a receptor, it's going to get stuck in the membrane. If there is some sort of Golgi or ER retention signal, it'll get hung and stay in the Golgi. But otherwise, if it doesn't have any transmembrane region or some kind of retention signal, its next step after Golgi processing, is being dumped out into the extracellular space. That's how we begin building the extracellular matrix. So this is very Golgi intensive. Also, not surprisingly, remember the Golgi is the site of all kinds of glycosylation. And the proteins that are in the basement membrane and the extracellular matrix in general, we can depend on them being highly glycosylated. So, we now have a structure that we can begin breaking apart. And not, inconsequentially, when we were doing our differential extraction procedures, which, just by the way, Tuesday's gels are looking pretty good. Thursday's gels look like hell. I don't know why. But regardless, we're going to have some nice data. We were doing this sequential extraction. SDS was pretty much pulling all of the cell material off of the basement membrane. And what you're left with is the basement membrane. And if you recall, the way we extracted proteins from that was with 8 molar urea. And we can begin to extract some of the proteins of the extracellular matrix. Some of them you ain't going to get out. So let's start taking apart the extracellular matrix. I don't know how many of you have either watched a concrete slab being poured or are in the concrete business, the construction business. A show of hands. Pretty quiet here in the room, but gender bias, man. We should all be low in construction, just the same way I know how to sew a button and I can cook. I don't know how to pour a slab though, so what the hell. But regardless, if we think of this, when you're pouring a slab, the first thing they do is lay down rebar, which is a little metal pole, and they got little wires, twist ties. They actually physically twist tie these things up, and they will make what looks like a little, a checkerboard or a screen of rebar. And you can oftentimes kind of, it'll be supported up by little metal poles or driven into the ground. And then you come back and you pour in concrete on top of that. Now, if you're making a really small concrete square, like a little decorative, stepping stone or something, you probably don't need to put any rebar into it. But if you're going to be making a piece as big as even like a sidewalk square, the pressure of walking and the pressure of the ground moving around is such that the concrete really doesn't have sufficient integrity, and it'll begin to crack and start to fall off. Now, if you put rebar in there, that rebar is actually serving as a support and keeping the whole thing from flexing as much. A similar analogy is going to be held when we start thinking about the animal extracellular matrix. Additionally, another thing, have you ever thought about when you're building a slab, how do they set up so you can put wood pieces on the top of the slab, the bottom of the wall, start building the wall? Have you ever thought about that? Well, I'll tell you, there's a couple of ways you can do it. You can either have a gun that'll shoot a nail through the 2x4 into the concrete, but if you're paying attention, often what they will do is they will have a number of rods sticking out of the edge of the concrete slab, and where there's going to be a wall, and then when it comes time to start building the wall, they'll put holes in the stick or the 2x4 plate, lay it down there, and then they can thread, lag bolt the whole thing down. We'll see that we actually have this kind of connection as well. So if you haven't done a big concrete pour and poured a slab, that's something for you to at least YouTube this week. Regardless, let's start taking apart our basement membrane. And the first part of the basement membrane is a glycoprotein known as collagen. Now, collagen is a really big protein, upwards of over 200,000 molecular weight. Collagen, you can find it in a whole range of skincare products, and I have to say that the application of collagen, particularly cow collagen, which is extracted from hooves and skin, to your skin is probably not doing anything. No, I'm sorry. And then particularly when they hydrolyze collagen and they sell it to you to put on your hair, it really isn't doing anything, but it's something to add to your shampoo and it's something to charge you more for. But collagen is a very long protein, and it does essentially form kind of a rod shape. And it actually is sort of coiled around. There's 3 collagen fibers coiled around each other. There are a bunch of different collagens. In fact, we can look at what the latest number is, but they're referred to as type and Roman numeral type 1, and we're up to like type XXIV, 24? I don't know. There's a lot of collagens. And what this means is collagen is a really big gene family. There are a bunch of collagen genes. So there are different collagen types, and they can be ranging from really, really tough to slightly squishy. One of the examples I always think about is collagen makes up the major protein in your tendons and ligaments. And if you want to get a sense for different collagen types, try this. There's a fair amount of collagen in your ear. Not really so much in the lobe, but in the slightly, harder, more rigid spot. So feel that, and then feel the back of your Achilles tendon, or whatever, feel your tendon, and you can sense that you've got some really different kinds of structures going on here. You have something fairly squishy to something pretty damn stiff. And these are different collagen types. So there are a lot of collagen types. But in general, collagen is going to form this 3-stranded cable. Now, one of the things about collagen that is really kind of interesting, and one of the reasons we have so many different collagen genes, is that collagen can be recognized at the molecular level, at the amino acid sequence level, fairly easily. So as you're screening through genomes, you say, that looks like a collagen. Specifically because, and this is the general notion, because the fibers have to lay on next to each other, a little bit like intermediate filaments, collagen has a stretch of repeating amino acids. Generally, it is going to be a stretch of prolines and lysines, and it'll be repeated over and over and over again. This proline-lysine repeat is sort of a hallmark of a collagen gene. Now, if you've been paying attention, proline and lysine are some of our basic amino acid residues, but more importantly, they can be modified. Because what we need to do is have a collagen fiber that's going to be really staying next to each other and stuck together. So these are not particularly large R groups sticking out. It does make for a very bulky protein chain. Additionally, there is a enzymatic modification post-translationally of the prolines and lysines in which we're going to add an OH group to create the modified amino acids known as hydroxyproline and hydroxylysine. What this is going to do is not only provide sort of a smaller R group so the strands can kind of lay in really close to each other, it also increases the ability of hydrogen bonding. Because these strands are not covalently linked together, but they are hydrogen bonded together. And if you are deficient in the hydrogen bonding of collagen, you can begin to see some of the kinds of effects or some of the structures that collagen is involved with, and you can actually, even on yourself, although I don't suggest this, do the experiment in which you can limit the amount of hydroxyproline, hydroxylysine, thereby limiting the amount of hydrogen bonding possible by ascorbic acid levels. Vitamin C is a critical cofactor to this enzyme process. So, all you got to do to find out what collagen is doing is restrict your amount of vitamin C that you're consuming. At least we as humans. Some organisms, they can produce their vitamin C on their own. This doesn't apply to you. So all you various birds and pigs out there in the audience, no. But for those humans, if you don't have sufficient vitamin C in your diet, you begin to lose the amount of hydroxyproline, hydroxylysine, and collagen being laid down, and you end up with a condition, of course, known as scurvy. It is vitamin C deficiency. And the symptoms of scurvy, if you ever read any of your pirate stories or sailing tales, you begin to lose structural integrity in that you bruise very easily. The myotendinous junction isn't formed very well, and you have muscle weakness and muscle tear. You begin to have loosening of your teeth in the gums. Scars begin to open back up. We need to keep a good rein on our collagen, and you need to maintain your levels of vitamin C because of this. So scurvy and the symptoms of scurvy end up being exactly the things that collagen is doing. That is maintaining tissue integrity. So, we were talking a little bit about cell-cell junctions and the ability of intermediate filaments to transmit this kind of stress. And wow, wouldn't that be horrible if my skin were to go pow on camera and I begin spurting blood? Regardless, I'm sure somebody here knows CPR. Collagen is also responsible for some of the elasticity in your skin. And we'll see here in a figure in just a moment. There are a couple of mutations that people can carry around with them. Not lethal, but not great. One is known as rubber man syndrome. And these are people that are lacking some of the structural elements in collagen proteins. They still have collagen, but they're not as tightly adhered, and they can really stretch out like that. A specific kind of genetic mutation known as Ehlers-Danlos syndrome results in what's known as the rubber man. Another condition that affects collagen that perhaps you've heard of is something called Marfan's syndrome. Marfan's is another mutation in specific collagen gene families. Marfan's results in a general kind of defect in a lot of the collagen extracellular matrix surrounding some of the major vessels. So people that have Marfan's are at tremendous risk of having like an aortic aneurysm, a blowout. People that have Marfan's also typically have some interesting phenotypes in that they tend to be very tall, fairly skinny, often a pronounced Adam's apple in dudes. It is thought, and there may actually be evidence now, that Abraham Lincoln had Marfan's syndrome. I actually went, during my postdoc, there was one of my colleagues, an Asian woman who was really, really tall for a Chinese woman and very, very slender, and she almost had an aortic aneurysm during her aerobics class. And so she was a Marfan's, and what they do is take a Teflon patch and sort of overwrap your aorta, and you're good to go. So you can get by having some defects, but it's going to have some issues. You're definitely going to have some problems with this. We're going to look at a couple of figures just because right now, and we'll see the rubber man, and we'll see how collagen is being laid down, and then we'll get into some more parts of the extracellular matrix. In terms of our analogy in the concrete pouring biz, collagen is rebar. We haven't gotten to the concrete yet. I don't know, dark, light, whatever. All right. here it comes. Wait for it. Our gut cells. All right, they actually are drawing them a little bit different than I am, or sort of we have the epithelial cells, and each one of these guys has their own lumen. Fine, we have to go up a little bit higher. But this is sort of a lower mag, okay, of the intestinal epithelia. And so we got the epithelia itself. This, of course, is being sloughed off quite regularly. You have cells underneath that are supplying the new epithelial cells. So then underneath this, the term connective tissue. It's another term for extracellular matrix. Gristle is not a particularly attractive term for extracellular matrix, but that's what it is. We've got a range of different fibroblasts and fibers. Here's some smooth muscle going on here in a couple of different orientations, so you can begin to stretch and twist and constrict. More connective tissue, lots of extracellular matrix stuff, and then another layer of epithelium, and this here is the one side of your intestines. So plant cell walls, yeah, pretty cool. And there is cellulose and pectin, turgor pressure. Wow, cool. Oh, actually, now we're on animals. All right, that was probably some kind of, that may be bone, actually. Regardless, here we got collagen. So we have the triple-stranded collagen molecule that then begins to build itself up into this larger collagen fibril. And then the collagen fibrils actually organize themselves up into collagen fibers. Actually, there's an interesting little point here. Collagen itself, happily, is really, really insoluble. I mean, that's why you can wash your hands. Now, a question is, something people were pondering is, how do you make an insoluble protein in the Golgi? I mean, this thing is going to be like a blob of, cow hooves sitting in the Golgi and actually secrete it out in the extracellular matrix. Turns out that collagen, as it is initially co-translationally translocated, has some globular ends on either side. And this allows the collagen to be soluble. The collagen is secreted into the extracellular matrix, and there is a protease, collagenase, specific type of collagenase, that clips those ends, and that causes the collagen now to become completely insoluble. So really, you're laying this stuff down as a soluble pasty stuff, and then the enzyme is coming in and clipping it and popping it and making it now into an insoluble material. It's a pretty cool little mechanism here that we got going on. And so you can see here's a cell embedded in the collagen fibers. And this is a scanning electron micrograph looking at tissue where we have cells here and then all of this collagen being laid down. So lots of structure going on. Well, here's the procollagen and the proteinase, and there you go. And so here's a guy that has got some mutations in his collagen gene. Don't you wish you could do that? And all right. So the next step is, how do we connect and link cells to this collagen matrix? And let's go back to the board. So collagen is our rebar. We're going to get to the concrete in a bit. The next step, though, is, partly, just a little, one of the reasons why we're doing it this way, is that collagen is pretty much all, it is a glycoprotein. These next proteins are also glycoproteins. The concrete that we're going to pour in is pretty much large carbohydrate, or a little bit of protein, but a huge amount of carbohydrate, what we call a proteoglycan. So that's probably the reason behind this order. That's the way the book does it. What are we going to do? All right, so the next step in building our extracellular matrix is the linker, the cross-link between the collagen fiber here and the cell here. We now have to invoke and develop the collagen cell linker system, the linker proteins. And there are a number of them, but there are two biggies that we like. And these are proteins that are also extracellular matrix proteins. Their job is to link the cell surface to the collagen fiber. And the two primary proteins are known as fibronectin and laminin. Fibronectin and laminin are two major extracellular matrix proteins. What they're doing is linking collagen to the cell. And so fibronectin and laminin, they have the responsibility of serving as the anchoring point for the cell. And if we think about this from a mechanical standpoint, there are a couple of things we can look at. Fibronectin fits the model a little bit better, so we kind of hit that one first. Fibronectin is functioning as a dimer, as a sulfhydryl-linked dimer. And let's see, it's a good color for fibronectin. It looks like this. You got a kind of a V shape going on here. So you then have here the disulfide bridge linking these two guys together. And the fibronectin molecule, we can expect a couple of different sites on it. First, there are sites that are collagen binding. And then we have sites that are the cell attachment sites. I mean, this makes sense. So we have collagen binding here and here. And then we have the cell attachment sites. All right. So now, what we can begin thinking about is, all right, this thing is binding to collagen. What is it binding to on the cell? And it turns out a really cool set of experiments determined what we call, or what was initially known as, the fibronectin receptor. The fibronectin receptor is a transmembrane protein, and we can kind of begin thinking about, all right, what do you want in a receptor that's going to interact with the extracellular matrix? The fibronectin receptor fits this bill. It's a transmembrane protein. Actually, it's a collection of them. And we can draw a plasma membrane here. Here's our collagen up here. And we got fibronectin binding like so. And the fibronectin receptor is a protein that is sitting like this in the plasma membrane. Actually, a bit of a dimer. This is a protein complex that's also known, in addition to being called the fibronectin receptor, it is a protein complex known as integrin. And on one hand, you're saying, why all these stupid terms? But see, I did my PhD on extracellular matrix cell surface receptors, and integrins were a big deal. And so I know all about integrins. Integrins are protein complexes that have the ability to bind fibronectin on the outside, and then they have a whole set of actin microfilament binding capacity inside. And so what this means is we've got a connection here that interacts with the extracellular matrix, passing through the membrane and then attaching to the cytoskeleton. This is now some serious business, because now we can have a cell land on an extracellular matrix, through integrin make a connection, and not only does it have a little tack point there, it actually transmits that information inside, and the cytoskeleton is attached here too, and so we now have this whole structure cemented in place and anchored in a way that is not just, again, a jelly-filled balloon with a piece of double-stick tape on it. It is a jelly-filled balloon with a piece of double-stick tape, and then on the inside we have all this tubing and filaments structuring, supporting the cell on top of this extracellular matrix. This is pretty cool stuff. And we will pick back up on it on Wednesday. There are actually a couple of interesting sites on the fibronectin molecule itself that end up being the actual integrin binding site. And the experiments that were done to demonstrate that were pretty neat. All right, I will chat with you again on Wednesday. Bye-bye.